aanwijzing niet. Oké dan. Goed, sorry. Ja. Nou, die.
senior author. Senior author.
Morning.
Morning. Oh, right, you set everything up.
Yes,
That's
I
great.
just checked. Oh.
It was fun working with Carl Christian because I just gave him an idea. He'd go away and write the paper and come back 2 weeks later and show me the paper.
He was like that all his life?
Yeah,
Oh, sorry,
yeah,
no,
yeah.
it's not this one.
Hi,
Let's see.
I'm Robert Oostenveld.
Bob Donner.
So,
I'm a colleague
another
of Tim, and
part
I'm
of...
going to be on the position as well.
Oh, yeah.
I'm going to
Very
the
good.
symposium this morning.
Let's see, this is the USB thingy, right?
Yeah.
Oh, the live stream is on, by the way.
Yeah.
So I think so. Should be.
So,
Please. So,
there's a lot of talks on there, but
so
it's the
I'm
most
sure-
recent one.
let's even put them in order. I could now even... Let's put the program in. No, let's not do that. Please, you know where to find it.
Donda's talk. There we are. Now, shall I just put it over on the
Yeah, just put it on the desktop for the moment. Morning.
Now, of course, that doesn't distinguish it from all the other Donda's talks this morning. But,
Right, you could name it Turner.
yeah.
Yeah, I did that just last week. I had called my defense talk, just layman's talk, and then I put it on the Ola computer.
Mm-hmm.
And every single day they've got people calling their PowerPoint layman's talk.
Yes.
So-
Yes. Yeah, one needs to think of these things. So let me just check.
Yeah,
Shall I bring it up and make sure it runs? One can never be too careful, you know.
We've got this screen. I think is a mirror of that one.
Mm-hmm.
So this is just your presenter screen.
Yes.
The more
Hey, there's a familiar face.
Ah, right.
Yeah.
I know you. That is kind of why I positioned him there.
How are you?
Awake. There is one straight downstairs, or one over there, or one basically everywhere. So, okay. That one's You can also just walk around until it's not necessary anymore. So down. Just straight down, and you can't miss it. Okay. Morning.
Okay. And
Looks lovely.
Hey, Natalia Petridou. Nice to meet you. So I'm going to be chairing this morning. So.
all right. It's quite a nice session.
I'm not sure if usual is the right word, but it does happen. So
Right.
go ahead.
I think it's a good idea to take advantage of the
Yeah, the fact that
fact that you have these people.
It's crazy. I was going right Laminar fMRI and all this afternoon, right?
Yes,
So,
we can all have a go at you this afternoon. We can all have a go at you this
yeah,
afternoon.
exactly. That's right.
Good morning. So do you want to test yourself or is it working?
I checked it.
Yeah,
That's
great.
right. So that,
I'm all sorted.
Okay.
my plan is to quickly introduce the general thing.
Great, yeah.
Should I try my computer or?
Yeah, you're right. Please, go. There's
to
the
make
UV
sure.
thing. There's a speaker's view, and the presenter. This one is the mirror of the thing behind you.
Yeah, I use
And it's not a
Max.
touch screen. Everybody tries that, but it isn't. Right. There's one question. It's going to be live streamed and all recorded. So is that a problem with you?
Live stream is okay. Yeah, no, it's fine.
Right. Second question. That we can only record from these screens. So is it possible to put it
It's
on
Keynote.
there? Oh. Okay, no, that's fine.
You should have given me a heads
No,
up, but it's
it's only yesterday evening that I realised.
no, sorry.
It was yesterday.
That's going to be a little bit too stressful to.
No, that's fine. That's fine.
So,
It was yesterday TG held to get the live stream on the roll.
Hey, Natalia?
Yeah. Hi.
Yeah, Nice to meet you.
Then we go.
Yeah.
So
Yeah,
I'm
that's
gonna
working.
be chairing and I'm gonna signal at 10 minutes. So that's when you have 2 minutes left.
2 minutes left? I thought we had 15.
10 plus 5.
Yeah, but that's talk or a question.
No, I wrote. It was very clear in the email. It was 10 plus 5.
You can't have the eyes give too many talks anymore.
I know. It doesn't work.
I can do the math. That's 5 is 15.
What's the point? If you want to put your presentation on here, that's
Okay.
Yeah, good luck. I'll be kicking and screaming till the 50 minutes is over.
probably. Yeah, that's the.
I guess.
I wanted it to make a 2-day symposium, but Liz was like, yeah, but we don't have the budget to have them stay overnight. So, like, all right, well, never mind then.
Okay, let's see.
Sorry?
Oh,
From here?
right, yeah, but you're from the Netherlands. No, yeah, you can put it.
So you're gonna introduce Natalia. I'm just gonna
Yeah, so.
say this is Serge.
So
He's
I'm
next
afraid.
speaker.
Yeah, well, I don't want you to eat more time from your.
Take a break.
Yeah, I don't do LinkedIn. I won't do
Is there a mouse?
If it goes over by 15 minutes, then.
Yeah,
What's
yeah,
that?
yeah.
Is there a mouse?
There you go. So,
I
yeah,
don't
I
know
hope
if
you don't
it's,
mind if I don't give you a 5-minute introduction and say your name. Because you're the first speaker. But
Yeah.
One in there.
The big day?
Good morning.
Oh,
Ah,
morning,
all the
hi.
way from Germany.
actually, which one in the UK? Yeah, right. That must be Tim's twin, isn't it?
It was
Also a PhD.
is it gonna do it? Yes.
You have a few minutes. Did you have a cup of coffee or anything?
I
Okay, is everything going okay with your church?
Oh, very smooth.
Yeah, okay. Well, we used to come in.
Up.
Yeah, no, it's very trying to make ethical stuff. Actually, we now have this chip card, which chip card is really awkward. I think to travel, I don't know what you do actually. You have to get one when you arrive in the Netherlands, right? So to be able to use it.
You can still like the one you can get like the one.
It's awkward. The thing is, you don't use that.
It's like
Good idea. Good morning, Lee. Yes,
Very good morning, sir. Nice to meet you. You too. Oh, I find it very nice to meet you guys today, actually. Particularly your remark about the camera. I can see you. I had to be pressing on this like people use any of the space and so anyway, it's quite awkward to get a seminar in you want to know first? We'll give you that. We'll try and give you that. Okay. Yeah, sure. Yeah, we talked about this. Yeah, I don't want to wrap it up. Yeah.
Sure. And do you just want it on the main desktop?
Yeah, just put it there. That's great. We are starting. Yeah, the dentist is always 5 minutes late.
Yeah.
So.
All right, it just went somewhere.
Yeah.
Very pleased to meet you. Well, I recognise you immediately as the twin of Tim, knowing it.
Yeah.
Yeah.
Time is
Okay.
30. Yeah.
Just log out.
Ja, je mag gaan zitten. Is dat terug nog? Je mag gaan zitten.
Okay.
Yeah. There we go. Then I'm going to press the big red button, and then we're recording, because it is well, the topic of the symposium is layers and open science, and open science also means that everything is going to be broadcast, if you don't mind, of course. Welcome. Very nice that you're all here. I'm going to keep this introduction brief because we don't have too much time. The occasion of this symposium might be my defense in this afternoon, but it would be a waste if we got the brightest minds on layers and, well, all you hear is me in this afternoon. So that's why I invited my Corona to speak here this morning. And that's the plan, even though they've got only 15 minutes and we've got 2 chairs for the first session. Janneke will be very strict on keeping time, that it does not exceed 15 minutes. And for the second session, David Norris will chairing the meeting. And then, without further ado, I would like to invite Natalia Petridou to the stage.
Thank you. Just give me a minute to start the presentation. Good morning, everyone.
And let me quickly say, computer.
All right.
Yeah.
Good morning, everyone. I'm happy to open this symposium today. And my presentation is going to be discussing what features of the hemodynamic response we can extract with Laminar fMRI. So, in brief, I'm going to be discussing temporal and spatial features and in relation to the vascular neuron organization within gray matter. But a bit of background before I get into the main bulk of the presentation on what gives us our bold signal. So I'm zooming in there in a small part of gray matter. Let's see if I can... Yay. A small part of gray matter, and what you see there is a picture of the vasculature that's present in this small piece of gray matter. It's a corrosion cast after it's a picture after the tissue has been washed away, so it's only the vessels. And with bold, we're measuring from the venous side, and our signal is coming from ideally microvessels, capillary bed, and smaller venules, and also from the larger veins in the cortex, the intracortical veins, and pial veins as well. And briefly, how this blood flow works is we have activity being triggered in... Can you see the cursor? No.
Is it on the other side of your screen? That's...
Oh.
Yeah, that was it.
Okay. So activity initiates in deeper layers in gray matter. We have blood flowing from the surface, the arteries into the capillary bed, oxygen diffuses, and then blood flows back up from the venous side towards the pial surface. So I'm going to start with that and discuss temporal features of the hemodynamic response that we can extract. And again, here you see the illustration of the vasculature. And on the right is curves at different hemodynamic curves at different cortical depths, starting the smaller one from the deeper cortex and the larger one from the pial surface. And I have 4 curves from gradient echo and 1 curve from spin echo, which gives us the response of the microvasculature, to which we can compare a gradient echo that gives us response from all the vascular compartments. And you can see, if you focus on the beginning of the curves, the first thing that we see is that deeper in gray matter, the responses are faster, so we can actually track the flow of blood towards the cortical surface. The same we can see with the time to peak of each of the curves. So as you ascend towards the cortical surface, they get more and more delayed. In addition to that, we can also see blood pooling across the vascular tree, which is again indicated by the amplitude of the curves increasing towards the pial surface, and also by the width of the curves as they get wider as more blood pools towards the surface. And other things we can pick up that I'm not going to discuss at all, basically, but I'm going to note is that we can see a bit of the initial dip that it increases towards the surface, and of course also the post-stimulus undershoot. This is a very short stimulation, so we can look at the impulse response. In addition to temporal dynamics, we can look at things in space. This is a visual cortex. It's a checkerboard stimulation, so very straightforward. I think this is easier for me. On the left, you can see a T1 weighted anatomy with enhanced myelin contrast in gray matter. And we do that so we can actually pinpoint layer 4, which is there, striae genari. Next to it, we have a T2 star weighted anatomy. It's all very high resolution, 0.3 stropic in this case. And we can again see the striae genari, but the point of this scan is to be able to visualize veins as best as we can. And the zoomed in panels are for primary visual cortex, V1. We have our T1, T2 star on the right, and in the middle panels is our fMRI. This is 0.55 istropic. It's actually high enough to be able to see some anatomical details in the functional scans as well, such as the striae genari, which is, for example, here you can see it. But focusing on the color overlay, which is a bold overlay, we see that it very nicely matches what we expect in the situation. We have our highest metabolic demand in the feed-forward layer, i.e. layer 4 striae genari, and we see that our bold nicely colocalizes with the striae genari. But of course, we can also see contributions from, for instance, intracortical veins or bile veins as well. So this is for the spatial distribution in a best case scenario, but if we look at the summary of the entire region that we activated, we get different kinds of responses, which we think depend on the kind of vessels that contribute to your signal. So I'm showing here profiles to illustrate this effect. Actually, the cursor is annoying because I don't have too much space to move it. No. Yes. Okay. So, white matter, CSF, and those are profiles across cortical depth that show a different behavior. And here we classify them arbitrarily according to their trend towards the bile surface. So we have a very strong linear trend, as we call it, medium linear trend, but the interesting one is the weak linear trend, which is the blue curve. And in this case, we can, this actually is similar, it's a summary of the slide that you saw before. We have a bump in layer 4, i.e. our highest signal is actually colocalized with feed-forward information in this case. But it's quite variable, as you see, and it's an illustration of how this trend is distributed across the cortical surface. It's a flat view of V1. And we have a variable distribution, basically, of this trend across the entire area that we activated. Again, likely due to the contribution of larger vessels, or not. You can also get negative bold with Laminar fMRI. It's the same data set as you saw before, only in this case, we are suppressing the cortex of interest. So we can do that with a hemifield presentation that alternates. So we are looking at the same locations in the cortex. So let's say here, only in this case we have suppression. And it's clearly negative. And again, the trends are mimic in a way what we get in the excitation situation. But if you focus at the blue curve, it's of course noisy. Should be careful how we interpret this data. But we get something that's a little bit different than in an excitation situation. And we thought that in this case, this represents a silencing of the cortex completely, which we tested with electrophysiology as well, just to be sure. But again, I want to be cautious when we interpret that. But this is some results for the same experiment when we use intracranial electrodes in humans in early visual cortex, and we compare it to the bold for the same locations. And we see that indeed we have the blue dots show you that we have a complete suppression in the case of the negative bold, while it's a sort of linear relationship in the excitation scenario. So, negative bold as well. And finally, other features we can extract is actual neuronal organization. And this is an example of a work that was triggered basically by post-mortem histology investigations, which is the panel of the left. This is a flat view of visual area V2 that's stained for myelin. And it has neuronal groups that are column-like organized. And in this case, I'm discussing two types of these groups. They're called stripes. And the alternating darker gray and lighter gray indicates the organization of these groups as you see them in a flat surface. On the right, we have our bald fMRI in vivo, in this case, and we can observe a similar pattern in V2 as well that is similar to histology. But the nice thing about this kind of data is coming up. And it's here. If you look on the left, I mentioned already about the myelin-sensitive imaging in gray matter, and you saw the myelin stain, where you can see those neural populations. So we can also see them in vivo, which is illustrated in volume view, if you will, on the lower left. If we now compare our fMRI and our myelin imaging for the same people, we can actually start saying something about the point spread function across layers of the bald response, which is a graph that's illustrated on the right. We have white matter, CSF, and you can see the full width half max of those stripes from the T1 imaging and the bald response, and they're actually quite close. And with that, I would like to thank you for your attention. It was very quick. I hope you have a lot of questions for me. And thank you very much for your attention. So I can continue talking. Well,
Well, I mean, I'm completely naive to this, but to what extent do you think we're going to start to be able to do this kind of thing in higher order areas?
I think it's possible to do immediately. The question is what paradigm you're going to do. How are you going to design your experiment? I think this is the art, basically, in moving to higher order regions. I think David has some experience with that one already.
Could you please elaborate on your negative bolds? Could you please go back to the slide? So with our own studies, we've seen something like that, things that go below zero. But what is exactly your interpretation of the inhibition?
Well, it also depends on what kind of experiment that you do. In this case, it's a hemifield stimulation or suppression, so the entire cortex, let's say the left part of visual cortex, is not receiving any input at all. Initially, we thought that we're going to see some suppression feedback connections, basically from the contralateral, from the other hemisphere, and maybe some increase in the signal in supra and infra, but suppression in the input layer. But we don't see that. We see a flat line. And that's why we tested it with, which means that there's nothing there. There's nothing happening. So it's actually completely silenced. And if you look now at electrophysiology, you have the entire spectrogram, all of it, going to zero or negative, even the alpha power. So it's just silent. I don't know how this is possible, but this is what the data tells us.
The work on V2 with the stripes, does what you're doing actually help to elucidate what the functional role of V2 actually is? What does V2 do to the signal? Why do we have a V2?
I think I will pass that on to Serge.
Why we have, I don't know. But I mean, one thing that I'd like to say about strike is the start of the functional specialization, right? So in exercise, of course, you have areas responding to motion, so there's a functional specialization. And that you see starting in group 2, so the stripes are actually, process of motion information. And the anatomical features kind of match, so MT is more myelinated, the stripes that respond to motion are also more myelinated, so the anatomical functional spectrum match. And it seems, why have a V2? It seems that the visual system is developed like a modular system. V2 is kind of a modular system. I don't know, you can speculate on that, why we have so many visual areas. I mean, theoretically, you could just have one. We have more that seem to be specialized with different processing. And if you look evolutionary, then, V1 is, V2 and MT is shared among all primates, but the other areas are not. So that perhaps when new areas develop, you develop a new module in a new area. So that's very speculative.
What I'm hinting at really is the potential for doing discovery science with these techniques. That's a really important potential in terms of.
Well, maybe I can make one more comment on that. I live in the visual cortex, right? But but we do have there a known structure of columns and lamina, and V2 is actually in V1, and so there's a very nice structure of orientation columns, orthodontist columns, and laminar. And Human Lee sort of proposed this, computational unit for V1. But one of the hypotheses is that this kind of structures is not limited to V1, that you might have similar structures throughout the rest of the brain. And that's a hypothesis. We have columns known in motor cortex, and now we show these structures in V2. We know also in MT there's these kind of structures, but one of the hypotheses is that you'll find these kind of structures throughout the rest of your brain, which actually links to these cognitive questions. I mean, what kind of columns do you have there, if you have columns at all?
There's also so much to add to that. There's also some evidence that actually separating into little columns is advantageous in terms of volume. So basically, those units that really do have a similar function, you want to group them together so that wiring length is shorter, but also processing time is shorter as well.
But it's striking whenever anybody looks carefully at myelin-stained sections of the brain or cell-stained sections of the brain, just how much any kind of columnar structure at a millimeter scale doesn't pop out actually. It really doesn't. It's really not easy to see. It's not obvious, except in certain very clearly defined areas of the brain, like parallel cortex in the rat brain, and of course primary visual cortex. It's just not obvious.
Actually,
anatomically.
If you look at anatomically, the orthodontist columns don't pop out anatomically. Right? They're not different in terms of myelin orientation columns don't really different anatomically either. It's really functional that they pop out.
But if you stain for cytochrome C, you can see.
But that's after you remove one eye, right? And so it's the remaining activity of the other eye. So there's some sort of support. That is a functional type of measure.
Which means you need to know the functional properties actually not right. Anyway, let's continue this discussion during the break later. So let's move on to the next speaker, Bob Turner.
Can I use this computer? I have to find a way to. Yes. Good luck with the cursor.
Right. I think this is mine.
All right. So Rob is going to talk about how we can structure, how structure can inform function.
The first thing I'm going to do is find out how the cursor can inform you guys. So.
This screen. This is
okay.
the menu over there.
All right. Okay. Thanks. Right. Let's see. Okay. So I'm going to be talking about on a more general level as to what's the use of getting down to this kind of spatial resolution, what can we really learn about this? So I'm going to be addressing the question of how do we model the brain in a mechanistic way, in a realistic way. And I'm going to mention the issue of cortical parcellation in this respect. And then move on to the question of anatomical localization and myelination. Myelination equals maturation, something very important to recognize. And how do we localize function and activity? How do we assess causal interactions? And talk about experimental strategies for layer-dependent studies. So if we want to model the brain mechanistically, we have to define the components of a brain at the system level. Obviously, we can't use individual neurons or synapses. There's far too many of them to make any kind of model. So if we're lucky, we'll be able to learn a great deal by getting to the level of the 450 or so deep brain nuclei, subcortical nuclei, and the 200 or so cortical areas that can be defined in a typical human brain. That number of now 650 components should be plenty for any kind of modeling and is possible to handle with very fast computers. But we need not only to define the components and define them in vivo, but we want to understand how those components transform inputs to outputs to make a decent model. So we have to have some means of estimation of causality between the different components of the brain. And part of this estimation of causality depends on actually knowing how the components are connected to each other. What are the structural and functional connectomes, and in particular, in this context, layer-dependent wiring. And of course, the $1,000 question is will we get adequately accurate predictions from the level of granularity that we can actually achieve using techniques which are non-invasive, like MRI? I would love everyone involved in this game to read such studies as Carl Craver's Explaining the Brain in order to understand better what actually you need to model something in a way which is satisfactory to the whole discipline of neuroscience. So now, talking about myeloarchitecture, the cortical anatomy. So fundamental points are neuroanatomy is not random. Neurons are there to controllably connect. Anatomical detail has functional importance. It should not be smoothed out with 8 millimeters of smoothing, as is typical in many analysis techniques have been developed, including SPM, which I helped to develop myself. We should be far more concerned about understanding microcircuits which are within cortical areas and macrocircuits between cortical areas, and recognize that neural activity drives myelination in a bootstrapping process. So understanding cortical anatomy is a matter of understanding how it gets there. Axons within a given cortical area appear to be myelinated in the same time frame. U fiber fascicles appear to be myelinated together. I'll comment a bit more about this later. Myelination is a crucial marker of a neuron's functional maturity. It's not usually reversed. And myeloarchitecture is accessible in vivo by MRI, which provides the key to comparing brains across individuals, and its distribution may classify important cortical microcircuits. So the question is whether its distribution can inform causal models based on layer-dependent fMRI. So here's a nice myelin-stained section from my lab in Leipzig, showing the radial fibers projecting out from the white matter. You can see them clearly here because the slices in the plane of the fibers, and the horizontal fibers, the bands of Baillet, which run parallel to the cortical surface, but typically random in their orientation within that plane. And you can see very easily here a nice boundary between two cortical areas where the myeloarchitecture abruptly changes within a distance which is much smaller than the thickness of the cortex. And this is common across the brain. This is just expanded so you can see these bands of Baillet and the radial fibers and of course the heavily myelinated layer right at the cortical surface, about which there's about 3 papers ever been published, which is mysterious and exciting because of a huge opportunity. Going back years now, people have been able to observe this layer structure. For instance, this stria genari here in this study of congenitally blind subjects who happen to have a stria genari. You can see layer structure in other areas such as V5 out here. This goes back to 2011 in a 7 Tesla scanner in Leipzig. So, classical myelin neuroanatomists describe well-defined areas of cortex with relatively uniform patterns of myelination, with harsh-sharp boundaries, hair-sharp boundaries, as Oscar and Cecile Vogt claimed over a century ago, which coincide with the architectonic boundaries. And the more recent work by Glasser and Van Essen, which is derivative of work which we did in Leipzig, also claim many sharp areal boundaries. Myelination is driven by experience. This implies that within a given cortical area, neurons have shared experience over their period of myelination and must therefore be regarded as having closely related functions. This is really talking about the functional parcellation of the brain. Patterns of myelination indicate important local cortical microcircuitry. Connectivity defines function. So now, how do I get back? I'm using the cursor. Sorry?
Previous. Right mouse click and then previous. Second one.
Nothing happened. Oh, there. Oh, down there. All right. I wasn't expecting it there. So, yes, myeloarchitecture, in my view, is the key to functional localization. We have, on the left here, we have the Brodmann areas, and on the right, the myeloarchitectural areas defined by Elliott Smith back in 1907. This is still one of the most useful maps of the myeloarchitecture of the brain, which tells you how little research has been done in the last century on myeloarchitecture. There's only a few hundred papers ever been published. It's time for that to change. Now we can see myeloarchitecture with MRI. For instance, this is early work shown at HBM meeting in 2010, showing primary motor cortex and primary somatosensory, primary auditory cortex, B5, MT, B1, on a cortical map of T1, quantitative T1 obtained with MRI in vivo. This matches the Hop map. He was in the Vogt and Vogt neuroanatomical lab. He made this map using cadaver brain sections, indicating the degree of myelination as darkness. So that matches rather well. And in the paper published by Glasser and Van Essen in 2011, they replicated this this result. One can do better if one takes these maps of T1 in the cortex and uses them to co-register a number of brains and then average across them. One gets very fine detail, and this is cortical layer by layer. This is using very high resolution, 0.5 millimeter isotropic MRI at 7 Tesla. And so using these techniques, we can parcellate the cortex on the basis of the cortical profiles, the layer dependence of the myelination. This gives us also a doorway into understanding the local cortical circuitry. It helps very much now to understand that a lot of these horizontal pathways are actually inhibitory. This is work of Micheva et al. In 2017, and they arise not from pyramidal cells but from basket cells, which are inhibitory neurons which basically control a fair number of neighboring pyramidal cells. So this is a door that's been opened by recognizing that MRI is looking in great detail at myeloarchitecture. So now we want to understand causal directionality. We know that from histology and animal brain research we can talk about the cortical layer specificity of input and output pathways for each cortical area. Experimentally we can drive activity in input layers, and experimentally we can observe the behavioral effects of activity in output layers. So if we can discriminate these layers with the spatial resolution of fMRI, we can then talk about causal relationships between brain areas. We can distinguish between inputs and outputs. Where we have top-down and bottom-up apparent separated spatially in a particular area of cortex, we can also estimate causal directions if we can devise paradigms which will either activate one or the other. The complexity of the cortical wiring is indicated. This is taken almost at random from a paper by Feldmeyer in 2012, rat whisker barrel cortex, just showing how rich and complicated the wiring is, but also indicating that one can discriminate input and output pathways. Once one has this notion that we can actually separate these experimentally with MRI, we can start thinking about the kind of paradigms that we might want to use, where you have layer-specific neural activity in the same cortical area. So, for instance, motor imagery versus actual motion. This is work we did in Leipzig in 2010. I'll mention very briefly. Visual imagery versus actual vision. And I think the work here done by Koch, which uses visual illusions, and the Knitzer triangle is an example of that. Auditory imagery versus actual hearing, cross-modal versus unimodal stimulation, top-down versus bottom-up attentional modulation, self-motion versus other motion in mirror system brain areas, touch experience versus touch observed. All of these offer opportunities of discriminating input and output layers in different areas of the cortex. I'm just going to mention very briefly work that, as I I did with Robert Trampel and Andreas Schäfer in Leipzig in 2010, which was a pioneering study. Didn't get published at the time, unfortunately. We used 0.4 millimeter isotropic structural images to identify an ambiguously specific cortical area of interest and to compute cortical contours. Use very high resolution fMRI. Use a task paradigm in which the different conditions each involve the same cortical area but with differing input and output characteristics. Average the bold contrast across subjects from the corresponding cortical area and layer contour by contour to get better contrast to noise. So what we did, and this was presented at the Society of Neuroscience meeting in 2011, high-resolution functional mapping in primary motor cortex and primary somatosensory cortex in humans at 7T, and it was published eventually in the Philosophical Transactions of the Royal Society in 2016. Here's another myelin-stained section from Leipzig. This is primary motor cortex on the left here, primary somatosensory cortex on the right. You can see how thick the primary motor cortex is and how full of myelin it is. And look at these lovely bundles of myelinated fibers going down almost to layer 2. And nobody has ever been able to tell me why they're there. And a contrasting myeloarchitecture in somatosensory cortex. So when we come come to look at, this is now a T1 weighted image, not a T1 image. You can actually identify primary motor cortex very easily because it's heavily myelinated. You then do finger tapping or finger moving or motor imagery versus rest, and you can see primary motor cortex lighting up in each of these tasks. This is with a 0.75 millimeter isotropic resolution, and these are two typical subjects. They're very similar data. This is the activity for the motor imagery task. Very unambiguously, primary motor cortex is involved, and there's a very recent paper showing that there's good reason anatomically for that. There are two types of pyramidal cells in motor cortex, and some of them are to do with preparation of motor movement. So what we did was to layer the cortex into 4 layers. The third layer down is mostly primary, the layer 5, which has the giant BET cells in it, is the output layer of motor cortex. And then we looked at the time course in each layer, and we found that in the finger tapping and finger moving conditions, we have a good separation of the time courses in each layer, which is characteristic. We have most bold contrast in the superficial layer and least in the deepest layer for very obvious hydrodynamic reasons. But in the imagining condition, we have the middle and deep layers are much closer together in their amplitude. The error bars in these curves are tiny, by the way, because we've averaged over so many points. Once we normalize with respect to the amplitude in that deepest layer, we find that the imagining condition is very significantly weaker in its activation compared with the other layers. So there's the imagining, there's the tapping, and there's the moving, the red bar we're looking at. And the conclusion for this is that we are really looking at the absence of output from layer 5 in the imagining condition. If there was an output from layer 5 in the imagining condition, it wouldn't be an imagining condition, it would be a motor condition. So, concluding, what we can gain from sub-millimeter resolution is we can identify brain components in living brain using myeloarchitecture. We can understand using myeloarchitecture what the cytoarchitecture is, and then we can assign input and output layers for each area. And then we can use techniques like cerebral blood volume mapping or some types of bold contrast fMRI to map pericyte activity to attribute causal function layer by layer. And I mention now, for people's interest, the recent work by Martin Lauritson's group in 2018 showing that pericytes do indeed lie in the right places to give you very detailed control of cerebral blood volume, and they're found throughout the cortex. And just to finish, I just want to plug the technique recently published by Gabby Lohman called LISA in Nature Communications, which is a much more spatially precise way of analyzing fMRI data. So thanks for your attention.
So while Serge is setting up his talk, maybe we have time for
yeah, you kind of elevate myelin to a very high place in telling something about function, but I kind of think of it as being like having a GPU in the area, right? In the sense that the highly myelinated regions are the most primitive ones in many ways, primary motor, primary visual, and actually the real challenging areas for cognitive neuroscience are the higher order regions, which tend not to have a very strong myelinization and are more challenging to look at. Would you care to comment?
Well, it's surprising how much you reveal once you start doing some proper time averaging, because there is myeloarchitecture everywhere. And going back to the work of Elliott Smith, he used cadaver brain. He didn't even have a powerful microscope. In 1907, he was using a powerful lens, basically, looking at fresh cadaver brain, slicing it, and looking at the bands of biaising with that primitive equipment. And with that, he was able to distinguish about 50 areas, many of them in prefrontal cortex, just in terms of visibility of the bands of biaising. So it shouldn't be so difficult. Yeah.
All right. So thank you, Bob. Serge Dumoulin from Utrecht University and Zoonosis. One from Laminar MRI about the brain. Serge.
Okay, thank you. I like to walk around, so I'm just going to try this one out. Does this one work? But it's working, right? Is it, no? It was a moment. There it is. Yeah, it's live stream, right? And I was told my Mac is not live streaming, so everybody's looking at the live stream. They're seeing it, or?
I think so.
Okay. All right. Well, thank you very much, and I'm glad to be part of this symposium. I was very excited when Tim invited me, and I didn't actually realize that he gave me a title, but I agreed to that as well. So I can't blame him. I could only blame myself. Looking back at it, preparing for this, I said, okay, I'm going to talk about what Laminar fMRI can teach us about the brain, and I will try that a little bit. But to be honest, I'll talk a little bit more about V1 and PRFs, which is really what I do in my daily life. Or scientific life, I should say. But I'm going to say a few general remarks. So I think most of you are familiar with these maps over here. This is the map from Brodmann, and this is really in the early 1900s, and really defined the different areas in the brain, and we still use them as a reference. And one thing to realize is that these ones were actually defined based upon difference in anatomical structures in the lamina. So this is the similar picture that Bob showed you before, based upon the cytoarchitecture and based upon the laminar difference in cytoarchitecture, they define these different brain areas. I show you here on the right, this is the myeloarchitecture, which is made by contemporaries of Brodmann, although they didn't publish such a nice map. But one of the ideas really is, if you can look at laminar, perhaps we can actually define these brain areas in the living brain, right? Rather than relying on the histology, we might be able to see them in the living brain. And we've been, for the last 20 years since fMRI has been around, we've been localizing activity based upon Brodmann areas, but we've never been able to see them. And we think they relate somewhat to the sulcal anatomy, but that's about 1 centimeter precision at most. Maybe a little bit more for early primary areas. So really, one of the sort of, I think, challenges for the future is can we actually identify these myeloarchitecture areas using MRI in living humans? And there's some evidence that we can see those structures. So this is an anatomical MRI. This is what you usually see with the 3T. But if you look at the 7T or ultra-high field, you see these structures appear. So this is this tree of genera. This is where the input of the eye arrives. And here, this is an extrastriate cortex. It's the lines of Ballaget, where we think it's the lines of Ballaget. Bob showed a nice picture of the real myeloarchitecture or cytoarchitecture. That turns out there's actually 2 lines of Ballaget. We can only see sort of 1. But this is very promising. So this is actually showing us, okay, we can see these structures using anatomy, and so perhaps we can start identifying these areas. The other interest, especially for cognitive neuroscience and for functional imaging, is the connectivity. So this is a basic schematic simplified diagram where you split up the cortex into 3 layers. We know there's 6 layers, and even those 6 layers are often subdivided. But if you look at those 3 layers, the granular layer, the central layer, this is where the information from the subcortex arrives. And this is also why in primary sensory areas, the granular layer is very strongly present. In V1, for example, this tree of genera is one of the strongest ones, one of the earliest ones to be identified, but in the motor cortex it's virtually absent.
So one of the ideas, so perhaps the laminar information can tell us something about the connectivity, and there's been a few studies also coming from here saying that yes, this might be possible. So can we, this sort of the second promise of laminar fMRI, can we reveal the information flow between areas based upon the laminar connectivity? I want to just highlight some caveats, so this is not as simple as that. So first of all, we have 6 layers where some of them are subdivided. You know, the input and output layers is the first synaptic connection, which a few milliseconds later it's transferred throughout the entire cortical thickness and between areas. So at the resolution of fMRI, you know, this is not just the first synaptic contact that matters, it's the whole circuitry that matters. And I'd like to sort of illustrate that, you know, to some extent, and I realized this recently, if you look at pyramidal cells, they're actually quite large and actually extend the whole, you know, the cross-section of the cortical thickness. So, you know, I should maybe not say this, but to some extent we're dealing with cellular resolution, right? We're imaging different aspects of the neurons, but that's to illustrate that even if we measure at the fMRI resolution, you know, the story I told you about the input-output is the first synaptic input, and this may not be what we measure at fMRI, the low resolution. The other problem is at fMRI we don't measure neurons directly, we measure the vasculature, and I think both previous speakers have shown the similar image before. The blood is actually supplied from the surface, and then it's supplied into the cortical depth and is pulled back up. So you're dealing with this vasculature, a mask really. And one thing that this mask does is if you plot sort of white matter here and CSF here, this is the cortical depth, that the amplitude will typically increase with as you go towards the pile surface. And this is not necessarily a neural signature, this is a signature of the vasculature. So we have to punch through that. So next, talking a little bit about my work in this field. I work predominantly with population receptive fields. So the notion is that every neuron or every neural population doesn't process the entire part of the visual field, but a small part of the visual field, and it's the population receptive field. And this is one of the basic properties of all sensory neurons. They don't process the entire sensory space, but a limited part of the sensory space. And, you know, we first sort of delineate where is that space, and then we can ask questions about how the neurons are processing it. And we, you know, build little computational models to model that. Going back to laminar connectivity, okay, how does this relate to laminar connectivity? So one thing we thought is that the population receptive field method can actually circumvent the problem that the intensity or the amplitude of the signal increases towards the pile surface. So if you look at amplitude measures, they're always going to be suspect in laminar fMRI because of this confound. And so we thought, okay, if we move to population receptive fields, the more signal we get, the better we can measure. I'm going to have to move then. No more battery apparently. So I'm going to go over here. So the one thing about population receptive fields, the nice thing is that, you know, the more signal you get, the better you are to able to characterize all the population receptive fields, but they don't become more eccentric or bigger or something like that. So measuring population receptive fields is a computational model that's independent of signal amplitude. So we want to look at the two laminar components. So, you know, can we tell something about the sort of ascending or the feed forward information compared to the descending or feedback? And when we look at the ascending information, this is work predominantly done by Alessio Fracasso. And if we talk about ascending information of feed forward, then the typical observation that we observe across visual cortex is that the population receptive fields get bigger. So this is, you know, the population receptive field size in degrees of visual angle. In early visual cortex is small by blue, and then later visual areas become progressively larger. If you plot the population receptive field size as a function of eccentricity for different visual areas, then V1 is the smallest, V2, V3, V4, and LO1. So this is kind of indicative of this hierarchy between the visual areas, even though we know there's a lot of feed forward and feedback information. So, but we like to think of this as a hierarchical system. And one signature of this hierarchical system is that the population receptive field size increases. So we wondered, does this also happen across the cortical thickness? Because the information arrives in the central layers and is then spread out to the more deeper and superficial layers. So the prediction would be that the population receptor fields would be smallest in the central layers and bigger in both the superficial and deeper layers. So Alessia worked on this for, I think, almost 2 years. And so I'm going to summarize it with just one slide. This is the population receptor field size. So this is the distance from the gray-white boundary. So this is the gray-white boundary. This is millimeters away from it. And you can see these this U-shaped curve, just like we predict. So the PRF size are the smallest in the central layers and bigger in the most deeper and superficial layers. Now, Peter Roelsema in the NIN across from us reanalyzed some of his electrophysiological data and published this as well. And this is neurophysiology. So this is monkeys where they open the skull and stick an electrode in. And this is the RF size in degrees. And this is cortical depth. 0 in this case is the central layers. And there's two nice things about this, really. Well, the first thing is that it's really similar to our data. And so you get a similar shape. And so this is really encouraging for laminar imaging, so we can actually recover some of the same properties. The other thing that I think is really interesting is that if you look at the absolute units, so degrees of visual angle, this is 2 degrees and this is 2 degrees. So if you take into account that between subjects, the PRF sizes varies with a factor 2 or 3, these values are very, very similar, taking into account that this is humans and this is monkey. So, two nice things. The shape is very similar and the absolute units are very similar. So I think this is very encouraging. So next, I want to say a few words about feedback information. This is work by Barry Klein. And you look at attention. So we can have the feed-forward information, but you can determine yourself where to focus attention on. And like I said, amplitude measurements are always a little bit suspect. So we didn't look at amplitude, but we looked at how the population receptor field changes with attention. This is a model that comes from Stefan Troy's lab and Reynolds and Heger. So you can model attention as an attention field. And you can model the receptive fields as a Gaussian. And if you model both of them as a Gaussian, and then you model the effects of attention as a Gaussian multiplication, to make a story short, if they're not centered and you multiply the two, then attention will actually bias the receptive field profile. And what you will have is that the preferred position changes. And you can think of it as, changing or reweighting the synaptic input, if you will, as a biological mechanism. But what this model predicts is that you get a preferred position change. So the receptive fields actually move, if you will, or bias themselves towards the preferred position. Now, this is what we found using also 7T, but at sort of regular resolutions. You can see these changes in population receptor field throughout the visual field and throughout the visual hierarchy. So this happens everywhere. So local attention really has a global effect. So it actually warps the entire visual field, at least between the 12 degrees of these attentional probes. But so local attention has global effects. So we actually said, okay, if we're looking at these changes in the population receptor field size across cortical depth, do we see any differences there? So this is the stimulus. The subject fixates these expanding rings. The subject does a task here on the left or the right. So there's actually no overlap between the attentional task and the stimulus. Here, just to show you some raw data, this is the eccentricity maps when they're tended to the left and to the right. And if you look carefully, you can already see that they're kind of shifted. This is just the data, in the voxel space that we get out, and we sort of remap them towards the plots versus cortical depth. But it's always nice to sort of raw data. If we talk about raw data, this is the time courses. And if you average them for attend right to attend left, then you see that this one is for attend right, this one is for attend left. And you see that the amplitudes are very similar. But you see there's a slight shift in the phase of the signal, and this is really what we're looking for. And if you plot that time difference or the phase difference, or we interpret as eccentricity difference, you see this for every subject reliably. Some subjects are a little bit bigger than others. But now we can actually look across cortical depth. So how does this position change actually happen across cortical depth? And this is the eccentricity difference across normalized cortical depth. So 0 is white matter and 1 is gray matter. And you see that the the eccentricity difference is actually biggest in the deeper layers and smaller in the more superficial layers. So you see that PRF attraction under the influence of attention varies across cortical depth. And this is a very cartoonish summary of how we think this might operate. So the information arrives in the central layers, the attention field arrives in the deeper layers, where you get the multiplication and the largest precision change, where this decreases across the cortical depth. So this is a very simple model that can capture the data. And to sort of sum up, I think laminar imaging is really promising because it can define, the different areas. This is the original definition of the different cortical areas. It can tell us something about the information flow between the areas, and I think our studies have highlighted that yes, you can actually see signals that are consistent with feed-forward information flow that's actually consistent with neurophysiology, and that attention actually, is actually differently employed across the cortical layers, and that is apparent when you look at position changes, but not necessarily when you're looking at amplitude changes. And last, I should thank all the people who've done the actual work, and in particularly Alessio, who worked on a lot of these laminar pipelines together with Natalia, and Barry, who did the attention work. Thank you.
Yeah, great talk, thanks. I can completely see why the PRF measure is unbiased by the amplitude, but I was not sure about the last part, where you have the position shift, basically induced by attention. Because aren't those position shifts basically the result of activity becoming sort of an increasing gain in one resonatopic location at the expense of another? And as such, if there's like a larger amplitude, would you then also get like a larger shift? I didn't really see how they were independent.
Yeah, no, sorry, I went over timelines really quickly. So the paradigm is that we don't actually compare attention versus attention. So we actually compare attention to the left versus attention to the right. So you always have attention employed, and you always have the attention field employed. So just locate at different positions of the field. So then if you do the multiplication, you'll have the same gain changes, and you still have the same sort of changes. You also predict changes in the PRF size, but you have them the same in both conditions, so they null out. And what you have left is, say, only the change in position really highlighted in this paradigm.
But the difference in attention, in both cases you have an increase in gain, but at different locations, right? In that sense, I don't really see how sort of respond. But maybe I'll read it.
I'm trying. I'm here.
Well, you can talk about it later.
Yeah, okay, that's it.
Maybe
unplug it.
I pushed that. Yeah.
If we get rid of this.
So maybe just drag this. Oh, it's in full screen. Here you can. It's on. Yeah. All right. So I just do this. Cool.
All right. So our next speaker today is Dan Sharoh, Director of Connectivity with Depth-Dependent Bolt. The floor is yours.
All right. And thank you very much. That'll be all. Yeah. So, all right. Normally, I get stuck rambling on about all of the background for far too long. But fortunately today, thanks to the previous talks, I can now ramble on about the experiment that we did, since you are now all expert in the laminar background and the anatomy and all the considerations that go with that when you're trying to think about how to put together a task to test with these functional measures that we were discussing. So we heard a lot also about what the use of this would be for, say, discovery science, or what the interest would be for people who are interested in more cognitive science. So that is what we do. And I think that this is specifically the potential to assess connectivity between different regions right across the brain using the cortical layers is one of the main motivators for cognitive science in general to try to apply this technique. So I'm going to be talking about our work with depth-dependent bold to assess directed conductivity, not in visual cortex exactly, but we move a few centimeters up and we deal with the occipital temporal sulcus. So we use a reading test, but can talk about that in a little bit. So yeah, we talked previously about a number of these studies already, but in recent years there's been growing evidence that yes, indeed, there is some, there is a strong spatial coupling between the hemodynamic response and the different cortical laminae that we can actually use to measure task effects in the deep layers or the superficial layers or the middle layers, depending on whether you're looking at feed forward or feed back. Specifically, that feed forward tends to focus on the middle layers or targets the middle layers, whereas feed back will target outside of the middle layers, so non-granularly. And we have mostly seen work in primary sensory cortices with this, so visual cortex, auditory cortex, motor cortex, and some work linking specific oscillatory frequencies to the different cortical laminae as well. But we go into a slightly more complex task where we look at language. So we do a single word reading task, right, presented in Dutch. We collected data on 22 subjects, and you can see a sample of the stimuli here presented. So the first two are real words. These would be salmon and battery in Dutch. The second two are pseudo words, so in red, right? And in the kind of fuchsia color, you have false font tokens. And the idea behind this is that the bottom-up information, the bottom-up stream, the sensory information associated with all of these tokens will be roughly the same, right? And there isn't really anything different in the visual stream comparing, say, the superficial or the real words and the pseudo words, right? Because these are all legal orthographic strings that Dutch readers will be able to understand. The difference is that the real words will have meaning associated with them, right? The real words are going to have experience-based information, knowledge on the participant's part on this, and they'll be able to draw from all sorts of different information related to the lexical, say, word frequency, even emotional experiences with the words. So we're not specific about what the top-down information necessarily is so much, but we do make assumptions that it has to do with the semantic content of the words, phonological content of the words, these types of things. So the idea is that by comparing that's too fast. Yeah, so the idea is that by comparing the difference between the real words and the pseudo words, we can actually have a strong effect where we only manipulate the top-down signal, where the bottom-up signal stays constant, right? And in doing so, we can then try to see what the effect of the top-down signal on the bottom-up signal actually would be, and then if we were successful in this, we were planning on going forward to assess the directed connectivity and seeing where this was actually coming from using GPPI, so the psychophysiological interaction analysis that many of you may already be familiar with. So, okay. The predictions that we had, and excuse the schematic, but I think it serves the purpose. So if you imagine you have two areas here, right, represented by these stained sections. And we would be presenting a real word or a pseudo word to a Dutch participant whose eye is here on this slide, right? The real word and the pseudo word would both hit the eye the same, right? No differences there. They would both travel back to these early visual areas, right? And then they would be taken into some unnamed lower sensory area, right, into the middle layers here, into the granular layers. Right. Now, from here, there's a whole mess of intrinsic connectivity that will be going on within the first region here, which would then project forward into a higher order region, and then ideally project back right into the original region. And so the idea is that the feedback from the higher order region to the lower order region would be stronger for the real word, which would be tafel in this case, which is a Dutch word. And that this would result in a higher amplitude, suspect or no, higher amplitude in the deep bin, right, which we could then use to test to see if we see a difference between these two words, right? And all this would be happening in the presence of a constant bottom-up stream. So the middle layer, we didn't really expect to be manipulating, right? So what we did was we identified a region in the OTS, right, using a contrast of the real, the pseudo words, and the false fonts to pick out the region that responded well to the, say, orthographically legal characters, but then showed preference to the real and pseudo words over the false fonts, because this is a functionally defined region which sometimes goes by the name of the VWFA, the visual word form area. But we're not being specific about what we actually call it, right? So we identified this region, right? We would see how well you can see that. Yeah, so we identify this region, and then we generate our surfaces, right? And we perform an equivolume parcellation. I used Tim's implementation of it, but following the Weinert method, right? And we then demixed the signal from the different layers, right, using the spatial GLM, which was also implemented by Tim. Thank you very much. And registration tools implemented by Tim. Tim did very much work. But you can see here on the left column of the longer figure, right, if you look in green, in the green box, you can see magnified what the area we're looking at is, right? And you can see in the middle window there, the middle cell, you can see what the mean functional image looks like. Right, and then on the bottom you can see the ROI zoomed into that. Right? So you can see that the surfaces follow along very nicely, and the anatomy image there is an inversion recovery EPI that we took for the sake of registration. Right? We can talk about that later if someone's interested. And you can see on the right hand side there. Yeah, and you can see on the right hand side going through from white matter to the peel surface, you can see how we did the voluming, right, with each voxel there that is yellow. More yellow has more of its voxel volume in a specific bin, and we go through the entire gray matter volume, and we assign each voxel to a particular depth bin, and this is what we use to actually look at the depth-dependent signal. So what we found was interesting. So if you, I mean that in a good way, right? That's a good interesting. So we had three gray matter bins that we were looking at, right? We had a deep, a middle, and a superficial. And as we talked about, we know that that's not the true anatomical picture, right? But we also know that bold, even sub-millimeter bold like we have, or sub-millimeter fMRI, is not going to be sufficient to really be able to resolve this finer anatomical structure. So we stick with three gray matter bins, and we know that, yeah, this is going to be integrating activity over, say, deeper layers, middle layers, and superficial layers, but roughly that the distinction should be preserved enough that we can tell differences in functionality based on differences in the input and output layers, right? So what we see here after testing for main effects of condition being the real word versus the pseudo word, right, for example, and depth, and then the interaction between the two, we see that the deep layer prefers very much the real word condition compared to the pseudo word condition, right? So you see this kind of, you see this effect where the deep bin will show stronger for real words, but higher for pseudo words in the middle and superficial bin. So it kind of reverses, right? And this is interesting because the overall signal for the pseudo word condition is actually lower, right? So there seems to be a specific deep bin.
It's higher.
Oh, sorry. The overall signal for the pseudo word that we observe in the non-laminar context is always higher, right? The real word is lower. So there is there's the assumption that there is a kind of inhibition that goes on, which is related to the top-down information in the say the semantic knowledge that you have of the real words, right? And so we can observe this here in the deep bin, which we predict will be the recipient of the feedback termination, the feedback signal, right? And seeing that the middle bin is actually higher for the pseudo words, the overall signal is higher for the pseudo words, we began to wonder if there was perhaps some kind of suppression effect going on that we could measure, right? With the to see if there was an effect on the middle bin from the deep bin, which varied with task, right? So we get to the interesting stuff, so I'll speed along. But we tested this in terms of a PPI, right, to see the effect of deep on middle. And we saw that deep and middle interact significantly during the pseudoword condition, but not during the real word condition, which is interesting considering that the overall signal is higher for pseudowords, but the deep bend signal is higher for real words. So we took this as some evidence of a suppression effect, perhaps, whereby the top-down signal acts to modulate the bottom-up signal and suppress the middle bend signal for the pseudowords compared to the real words. Right? But I'll go through this. Yeah, I'll skip this one. This is the very interesting slide that I wanted to share with you guys today. So if we take the deep bend signal and we take the middle bend signal, the time courses that we extract from this slide here, right? We take time courses from the deep bend and the middle bend, right? And we create interaction terms with the tasks to see how they interact with the rest of the brain. We find that the deep bend almost surgically targets areas related to lexical retrieval, areas in the core language network, right? So you have posterior MTG and MTG here. And this is the only two clusters that survive correction. It's only on the left. The middle bend does not show any clusters really surviving correction, and it does seem to show preference for pseudowords, but we could talk about that later if you're interested. But the I think fascinating thing about this is that it seems that with gradient equibold you can actually disentangle very spatially localizable signals, right? And the GPPI appears sufficient to regress out task effects enough to actually let you observe the interaction between the middle bend and the deep bend in different areas of the brain, even though those regions are so close together. Right? So this is a very positive outcome from our work that we look forward to exploring in the future. So I guess we're done. And I thank you for your attention. Yes. Tom.
Yeah, it's more of a clarification question. Maybe it's remedial. So, as I understood it, one of the things that spatial GLM does is kind of costs you space, right? You go from a signal that's like voxel by voxel, and then you sort of turn it into a GLM, which is giving you each layer as a separate time, and then am I talking?
Costs you space.
Can you comment on the spatial specificity of the findings? As I understood it, one of the things that happened when you put everything into this big spatial GLM is that you lost the ability to make spatially specific claims. Your last slide.
Well, you gain the ability to make claims going through the cortical volume, right? You lose the ability to make claims over, say, the surface, right? Yeah.
Okay, so then how can you elaborate a little bit on how you've ended up with this very fine, detailed spatial map that you showed at the end?
Find the spatial map at the end. This one. Okay. Oh, so this is a the GPPI at the group level. So this is the group level map. This was done in normalized space. So I took so for every subject, the different bins always stayed in native space, right? And when I conducted the group level analysis, it was always to compare the bins and the native brains. So we predicted activity in the normalized brains, right, using the native space data, right? So that's why it looks. So this is a template map, right, that I had normalized and aligned every subject's brain to, right? But the actual measure that we used for the GPPI, right, the seed regions themselves, those came from native space, right? So that's why it looks like this. Yes.
The interest of time. Let's continue the discussion during the break. There's going to be a break now, and we're going to continue at 11.
But that looks beautiful on this, but that's an average. Now, how did the individual subjects look?
Yeah, so
I mean,
that is
something which is very rarely found is whether anything said about the average actually applies to any of the individuals.
So this
the thing is, mechanism only works in individual brains.
Indeed. Yes.
You know, only an individual brain has a mechanism.
There's no group
and-
mechanism, right?
there's
Yeah.
and this is something which is escaping the whole field of imaging neuroscience and distresses me greatly, because it basically throws away neuroanatomy. On average, you lose the detail.
Yeah.
Unless if you go as far as getting myelin maps of the cortex, references to quantitative T1 maps, you can use those to normalize brains across a group.
Yes.
In a way which is realistic in terms of the actual functional neuroanatomy. And then you might just be able to say something about mechanism on the basis of average.
Yeah.
But otherwise, I think you're averaging over what might be completely disparate spatial organization.
Yeah.
It's just by chance you get a few little bits coming out of which is significant.
So the individual subjects are here, but you're not talking about the non-connectivity stuff. You're talking about the GPPI at the end, right?
About the maps, yeah.
You're talking about this, yeah. I agree. So there are a couple different analyses that I tried with this as well to avoid or to try to get more at the individual level response, where I would do it with ROIs defined in every subject, which is not anywhere near the specificity that you would like to see, right? But I still think it's better than this. The thing that the whole brain, I think, does well is it removes all bias that I might have in picking out the whole brain regions that I'd be interested in myself, right? Because so in every subject, right, we have this defined.
That's why we defined it in the first place. At the time, in the mid-1990s, there was a big battle with people who were very happy with picking out regions of interest. Per Rowland was famous. And SPM was devised in a way to try to make a level playing field and to prevent people putting their fingers on the scale.
Yes, everyone uses the same way.
And uses family-wise error as a fairly stringent assessment of But really, it made a fundamental mistake of smoothing the data before it was statistically analysed, which meant that you could no longer ever get back to a correlation of functional activity with neural substrate.
Right.
And so you could end up with maps with very tiny areas of activation and so on, which completely
Hey Bob, good to see you. You right?
Yeah, I'm fine, yes.
He's concerned about something.
You're still retired? Are you planning otherwise?
No.
You're planning a big comeback.
Next step is death, I think.
No. It's not going to happen anytime soon.
It better not. But, we're just talking about that map at the end, which is a PPI. But that's an average. I'm just really wondering how well it was replicated on individual subjects. And, I thought it makes it far more convincing if you can show that something which you might propose in mechanism actually is operating in an individual brain.
Yeah, and equals one. Yeah. I mean, if the starting argument is that you need to get towards finer-grained characterization of effect, right? Then doing it in an individual subject would be nice.
Because then you can tie it to neural substrate and what you know of whole activity.
Yeah.
Though with the GPPI technique, though, I don't know that it would be possible to do it with a single subject.
I see. I
mean, whatever you. I mean, basically, you're just using subject as a sampling space to increase statistical power.
Yes.
Right?
But
and you can do it in many ways.
No, I could take it in one subject, right? Collect data in one subject repeatedly or something like this.
Across time.
Yeah. You never know whether it will increase statistical power. You can only be sure of that if you know that every subject, once you've normalized them to SPM, whatever, these areas lie perfectly on top of each other.
Sure, but I mean, you could for instance, just look into a sliding window type of analysis. And so that's precisely what we do with Eric now, moving his ICP stuff, his instantaneous connectivity translation, towards individualized ICPs.
Yeah, yeah, but that's extremely fast acquisitions, right?
No, that's not even extreme fast. I mean, that's just done at multibanding. But the thing is that by not having the cross-subject level of variability, which is the overriding thing for most of the stuff that you do, right? You actually already gain a lot of. I mean, you then just need another dimension from which to draw the confidence intervals.
Yeah.
And that can very well be time. You don't need that many observations in time.
Well, yeah, and most of the difficulty we experience comes from the inter-subject variability.
Exactly.
Right.
Exactly. And if you don't have that, that already buys you a lot.
Yeah.
Yeah, exactly. Explained variance is good. Unexplained variance is not.
That's the problem, the anatomical variance across subjects.
Yeah.
You don't have another index, another marker that has an explanatory variance. So,
well, you could do something like we do in the HGP with the multimodal surface alignment, where you move to another modality and explain variability in this modality.
I tried something like that.
Mapping T1 in the cortex and then using the T1 in the cortex as a means of aligning brains. That helps a good deal. Make sure you compare like a lot.
Well, the alignment on the individual subject level is challenging enough. Right. Are you talking about across subjects, or are you talking about just making sure that you have the subject anatomy aligned to?
No, you can do it across subjects. You have to inflate the brain and slide the T1 map around until it maps.
Yeah.
I showed it.
Yeah.
That is worked by Christine Tardy, who's now professor working with 70 there, so I'm looking forward to great things from her. But that's that I think you have to get bloody good data to do this. It's true, 7 Tesla data. But it's definitely doable. And it's worth doing because you then are comparing something at the level of the neural architecture.
It is an interesting question how similar this architecture will be from our perspective once you get into higher order questions about language, for example. Because it's not uncommon in say, just at a more superficial level to think of it like this. It's not uncommon for the region that I pick out in every subject to be in almost a completely different place. Like if you look, this is functionally defined, but you can tell like these are very different brains, these are very different places, and to expect to be able to realistically align these and have the regions be similar in size.
One of the subjects in my 2004 study in Japan on speech and song had on bilingualism. It was Cathy Price. One of the subjects happened to have all his language in his right hemisphere. And if we hadn't looked at the individual data, we would never have known that we'd thrown in an enormous amount of variability.
It is important to check lateralization for language.
What was the consensus now? Are we going to start later?
I assume we have to, right? Like, I think we should be starting now, actually.
对那个 BLA 的那个行为和 local 行为，因为前两天跟
trying my best to not ever talk about predictive coding actually.
Yes, I know. But it's already in mind.
There's
这样的话你就没有效率，没有可思议。
I like the
这个就
distinction they make between like attention and different types of
影像
feedback
上面还有老师的
because from what I can tell, there's supposed to be an expectation that attention will affect players differentially compared to key players, and that different sources of information they affect key players, and that works well for our work here where we always find stuff in key players to measure gradient, but it's convenient.
一个病人吗？5个，太老
I don't think I fully get it. Probably I'm not. I'm just not on Laminar.
Human road study.
Yeah. I mean, well, it's like technical.
这做太多了。
Sorry.
Well, it's almost. I'm gonna get a coffee.
Yeah.
In the UK, there's a law which says you have to license.
Zo'n enorm gat en dan jij in de post.
Can you start? Sorry?
No.
That works. We need some assistance in dealing with the cockpit here.
Things are just
You want to use
too advanced.
You going to run it from your own computer?
I could also pull it from.
No, you can do it quite easy if you want to.
Yeah.
If you've got HDMI, yes, you've got HDMI.
Oh, okay. Yeah, this is going to be your phone.
You have some.
So how are you doing? Been a while. How are you doing?
Good, good.
You need some input.
Yeah.
Should go there. That's showing signs of life now. If you just tell it to export the signal first. Oh, yeah, you took it out.
Yeah.
That should
now somewhere?
Yes.
Okay, we have various illustrious guests who are still downstairs, so I suppose I'd better try and get them up here before we start.
No.
Right, well, now for something completely different. Or not. Open science. And we will, as time has progressed, Tim has developed from not only providing countless useful tools for laminar fMRI analysis, but to a kind of crusade to make open science a reality. And this is also reflected in the second half of his thesis, and we have a really nice collection of speakers for the second session today, and it's a pleasure to welcome someone else who also bridges open science and laminar fMRI in many ways, and that's Pierre-Louis Bazin, who is now in Amsterdam, was previously in Leipzig. Right? Okay, thank you. The floor is yours.
All right. Thank you very much. Thank you for the invitation. So after all these very elaborate talks, I will go very, very light on some, maybe more of an experience on open science and how it can be a lot more, less straightforward than you would expect. So it starts, of course, with high-resolution MRI, 7 Tesla and above. What we have is beautiful contrasts. Now we can really push anatomical resolution, bring quantitative measurements, and do fancy things like looking at intracortical contrasts. So those are great promises, but there's also great challenges, and those are, well, increased anatomical resolution, quantitative contrasts, and the fact that we want to see intracortical contrasts in those images. So basically what we have here is very, very advanced MRI. And when I started the Max Planck back in 2011, none of the processing tools worked. So we could get those images, look at them, and sigh. So that's where I started working quite extensively in building a toolbox for this kind of purpose, so-called the CBSTools, specifically to go through all the steps to do what people do normally with things like FreeSurfer or SPM or FSL, just getting the images into structures, labels that you want to use, go into cortical laminations, measure depth, and so on. So in detail, so it's a toolbox that has been designed specifically for 7 Tesla data, so thinking about high resolution data, high numbers of voxels, working in spaces like 0.4 millimeter isotropic without spending weeks, and then looking at quantitative contrasts, which are something that you wouldn't see typically. Well, it's changing a little now, but that you wouldn't have seen typically at 3 Tesla or below. And then building tools that are related to the 7 Tesla applications, such as layering, cortical profiling, and co-registration of surfaces with high accuracy. And so at the time, I thought I had a pretty good system. building the software on top of existing open software. So what I did is I took the NIH-developed software called MIPAV, which has a very nice user interface, lots of formats that are supported, and the GIST system, which provides on top of that some pipelining information, and then added a special set of tools to go with. So that way there's shared development with a lot of different groups. Everything is open source and available, and there's a lot of resources that I could pull and getting that like this. And then there was also tools from different labs that would also have the same system, so you could get a very nice ecosystem. So at that point I was very happy and stayed happy about this for a while, thinking, well, maybe it is a perfect solution. But no. Unfortunately, people are lazy, me included. So one thing that is very difficult is to install software that depends upon other software that depends upon other software. And that then became a major hurdle. Also, with that software tools, you could encapsulate other tools, which is great. So you can just increase your ecosystem. But it's not that easy, so you need someone with specialized knowledge to do that. And the third part, which I found very amusing, was, well, it's using Java. Java was really all the rage maybe 15, 20 years ago. And now it's just the worst language you can think of for everything. It's still efficient, cross-platform, but it's really not popular, and people just don't like it. So at that point, I had a very interesting experience. I got a lot of free advice from various people. Some people told me, well, you should really create a SPM toolbox. Other people told me, well, you should just do a standalone set of scripts that work with FSL. Others say, well, you should just do something that works with a GIFT format, and then we can put it on the HCP workbench. And other people saying, well, you just wrap your GIST modules into Python and then put them in NiPipe. So basically everybody was telling me the same thing. Can you please make your software available in our favorite set of tools? And so that's in the end what I ended up doing, and nowadays the way to do it is to go towards Python, which, well, is highly popular. So because it's popular now, all the cool neuroscience tools are there, and there's a lot of online community. And that's actually a big difference, because now I have some problems and bugs in the way I've run my program. And then I get an email from someone from a different lab who says, hey, I found a bug there. Here's the fix that I made. Can you just have a look? And then you can check on it and be happy with it. So that is very different from, hey, I can't install everything. Please help. And to get there, I couldn't do that on my own because I'm an old guy who does Java programming. Nobody knows what it is anymore. So I needed to enlist the help of some very skillful neuroscientists who happen to be also Python wizards, Julia Unternberg in particular, and also Christopher Steele. And I took also opportunity of open science and open software initiatives, such as BrainHack and Google Summer of Code, who gave us a little bit of free space and free time to just drop everything else we were doing and focus on the code, focus on getting things figured out. And the first step of that we did during a BrainHack session was to figure out some Python wrappers for Java that would work without using a different type of Python that would not be compatible with the original Python. The nice thing with that is then you have continuity in the code and everything. And then with that, Julia really just took 3 months off with a Google Summer of Code and then just went through and added all the features to make it available on GitHub with a documentation that is automatically generated, adding with other people's help, automatic testing, Docker containerization, all sorts of bells and whistles, and all these things that we really want in the software from the user side. And of course, at the end, of course, a cool logo, because we need one. So this is kind of the state we are at now. We have a fully functional Python package behind the scene. There's Java, but you don't have to know about it. It's okay. And the nice thing is now you can interface that with NiPipe, with NiLearn, with anything that starts with Ni and has Python in it, and things that also don't have Python in them. I heard it's possible. So that's about it. I just wanted to share a little bit that story that making open tools is not just enough. Just bringing your code, putting it there, is not going to go necessarily very far, because there's a lot of things that come in the way, things you think were useful, like user interfaces. They can be actually cumbersome. Competing tools, they actually want to work with you and compete with you at the same time, but so if you don't let them do that, then you cannot really compare. And the last thing I found was very puzzling, but it's interesting to think about, is that you really have to follow fashion, because the fashion comes with online communities, with infrastructure in software. And so right now, of course, the fashion is Python, and it's all cool for people who started with there and feel like, well, of course, that's the only way. But maybe in 10 years, it will be Julia or Ruby or whatever. And then we will have to think about how we translate our tools to make them still relevant many years after they've been started and developed. And with that, I thank you for your attention.
So we've seen fashion is important not only in dress but also in software. Questions? No questions? Bob.
Would you say that user interfaces are today's most perfect exemplification of the old expression of Procrustean bed?
I don't know that expression very well. You detail it.
Procrustean was a mythical person who used to torture his captives, but he had a bed of a particular length, and if his captive didn't fit on the bed, he cut off their feet or head.
Yep. Yeah. We do have that not only with so user interfaces have been that way. At some point, the web to the internet 2.0 has been also very strong at imposing very specific look and feel, and everybody had to do it. And now there's all sorts of problems also with different kind of standardizations as well, which are interesting. But if you want all your datasets to work with the HTTP Workbench, which is a beautiful tool, you have to go through the HTTP systems, the HTTP pre-processing pipelines, the HTTP tools, and so you are also stuck within that microcosm. And if your data is very different, especially at 7T, we see that all the time, every new data set is different, then those very standard approaches become sometimes a bit more cumbersome than they are actually helpful. And so it's a balancing act to try to maintain both, I think, because they're both valuable.
Great. The first comment. Yeah, then, so how does it work if somebody wants to contribute to your code now? Do they do it in Python then, or?
They go on GitHub and do a pull request. That's the stuff that people do now. There's Python and there's Java code. So if they want to code in Java, they can. But a lot of the things are now, everything is in Python, and then called some Java for things that require computation, because Python is not really good for that in itself.
Okay. Last No, not the last. Well, we have various questioners all wanting to ask.
People are warming up.
Tom, you're yes.
So then, I think it's really interesting to bring up this concept of fashion. But then, so I kind of, as a non-Python user myself, I'd like to know, like, how robust are you to the inevitable Python going out of fashion? Like,
well,
what will you do, and like how do you see this tool in like a future incarnation when people don't like Python anymore?
So the nice thing is now I've been working through the process of building wrappers on top of something that is already antiquated and out of fashion. So I can do that a different time, and it's not a problem, hopefully. And hopefully now that there's a big Python community that will be crying because they're not cool anymore, they will try to build bridges to automatically bring Python code back into whatever the new thing in fashion at the time.
You end up with like a sort of gobstopper model then, where it's like layer.
Yep. Yeah, it becomes a bit of an onion. But you can, of course, so you can do wrapper upon wrapper upon wrapper. I mean, I did try to do gist, so take the gist modules that are wrappers around my code and then wrap them into NiPipe so that they can be pipelines inside of pipelines with 3 different language programs. So that works, it's just silly.
So then how does that relate then to the initial problem you had, which is that people don't like things that depend on other things that depend on other things? Because it seems like that's basically an inevitable consequence of this kind of approach, right?
Yeah, so one of the nice things is when you go through that process, you have to lean down whatever your core code is. So right now my Java code is portable to pretty much any kind of wrappers. And I would basically take our wrapper and not wrap it around the Python, but around the Java itself directly. Because that is not going to change, and it's very simplified because it had to go through that process once.
If we can have a very quick question from
and also as a follow-up. So, say you were designing everything from scratch, then you would still go with Java and whatever the most popular scripting language around, just wrap it.
No, right now I've heard Julia is the new thing for doing like real computations. So I would look into that.
So in implementing everything from scratch with Julia. So
yeah.
Not as a wrapper.
Why not?
Okay, that's a very concrete answer. So thanks once again for a really nice talk. And now we come to the question which terrifies every cognitive neuroscientist. Could another lab reproduce our results? And speaking will be Umut.
You're an old hand here.
Can you? Oh, here. Okay.
No, you have to drag it over to here. First of all, open
but
it.
That is...
No signal, no replication.
Right. What do you want? I want to open a Chrome, but it's here. Okay.
It's decided to open it on the window which was turned off. That's why you now have to.
All right. Right, hello, everyone. So I too would like to start with thanking Tim for the invitation and also for choosing my title for me, and kind of also in a way the contents of the talk, kind of showing his indeed as David put it, crusade for open science, recruiting new members as he goes along the way. Anyway, so I will try to talk about could another lab reproduce our results, but mostly I'll focus on open data, because that's what we're trying to push ourselves in our lab as well. But first of all, I'd like to say that, sorry, this. I work at the AI department here, so just other side of the campus, and we use mostly deep neural networks, deep learning, artificial intelligence to analyze neural data. Hence, the type of data that we use is quite unconventional in a sense, which I'll talk about in more detail in a minute. But so we need very long specialized data sets. Hence, it gives the question: can other labs reproduce our results? Another context, basically, because most of the time it's more about this question. I believe it will be more about whether people can reproduce the results. The methods people will ask this from a methods perspective, but for us, the question becomes: can we collect, or can others collect this very sophisticated data sets to be able to reproduce our results? So, okay. So before all of that, let me just briefly talk about what our results is. So what I just summarize two studies also. I was just thought it was coming from me, so I was like, that wasn't intentional. So, as I said, well, I haven't said that yet, but we're interested in the question of how the human brain represents the features of its environment. So, the field of neural coding, to be exact. And to answer this question, we use computational models of brain function, which I mentioned we formulate with artificial neural networks, deep learning, deep neural networks. And we validate this on massive, these models on, by current neuroscience standards, massive fMRI data sets. So, and this typically basically means that for those of you who are not really familiar with artificial neural networks, these are computational models which can be trained from data to perform tasks that are of interest to us. For example, certain cognitive tasks like object recognition, or planning, action, whatever. And then by fitting these models on our fMRI data, we can try to find the connections between such models and the human brain, for example, to kind of then make inferences about how the human brain is solving such tasks that we trained the initial models on. Right, so, and very quickly to studies to get, give a bit more context. For example, a couple of years ago, what we did was to train an artificial neural networks to recognize objects in images, and then compared the, in a bit hand-wavy way, the behavior of these networks to different regions in the human brain by, after they're trained, showing the same set of stimuli both to the network and to our participants in an fMRI scanner, and kind of trying to fit a model which maps the networks, the internal activity of the network to the activity of the brain. And once we did that, we found out that, can you see my cursor perhaps? Oh yeah, okay, I can. So this is a flat map of the visual cortex of the brain. And our model was a deep neural network, so it had different number of layers, 8 layers to be specific. And once we kind of mapped this onto the cortex, we found that different layers of the model maps, systematically. There was systematic mapping between different layers of the brain and different regions in the visual hierarchy, such that the lower layers corresponded to the early visual areas represented with blue here, and higher layers of the model corresponded to the deeper downstream areas. And this results, this representational gradient, as we call it, was reproduced, not reproduced, extended, so I shouldn't use that word because that's a special word for this talk, but extended for other stimulus modalities and brain regions like the dorsal stream or the superior temporal gyrus for example, audio, music recognition, etc. And so once we have a model for the brain, then we can also invert these models, these deep neural networks, to do stimulus reconstruction from brain responses. So, and this is just another example of such a study. Here we again use a deep neural network to model the brain responses, the transformation from stimulus to brain responses, and then just use some probabilistic inference to invert this model so as to, from brain response alone, given the, so the brain response of a particular stimulus, just reconstruct it from this response. And here, the first column and the last column, just a symmetric image, you can see the stimuli that were presented to two subjects. And I think this subject is the chair of the manuscript committee of Tim today, so Marcel, who unfortunately couldn't be here. And then this, I am not sure, but I think that's Yağmur, another researcher from our lab. So, and the middle columns, these are the reconstructions from the just brain responses alone. Anyway, so yes, so I forgot to add the animation. So I want to build a little bit suspense to first say, could another lab reproduce our results, and then click yes. But anyway, so but my answer is yes and no. I already tried to allude to this at the beginning. Well, not yes and no, but yes, but basically. So so methods-wise, yes, it's given the data other groups. So also when I say can other labs reproduce, could another lab reproduce our results, I also mean that in the sense that not just can others reproduce our results, but can we also reproduce the results of other labs who are basically working on the same problem. So, and let me start with the yes part, then I'll move on to the but part. So. Yes, and the initial studies, basically, which use deep neural networks to study brain responses, etc., were done on the monkey. Were done to so deep neural networks were used in those early studies to predict the responses of monkey inferior temporal cortex. So basically, all of these results that I've been talking about, and what came after that is kind of like can be considered in a sense a reproduction of the this earlier studies which use DNNs with monkey IT responses. And then these results have been extended into the human visual cortex. So if you talk about this representational gradients, which I showed which I showed 2 slides earlier, so they too have been reproduced and extended in different regions of the brain. For example, with videos in the dorsal stream, with audio in the superior temporal gyrus, and also just the same results, but in a with slight twists and different slightly different architectures. Basically, the point I'm trying to make is yes, it's really easy, at least in our field, to reproduce these results. But still, I'm getting to the more interesting part in a minute, though. But also, let me say that these results have not only been reproduced in fMRI, but also with MEG. So in fMRI, we showed, for example, this gradient here in space, right? So by mapping the deep neural network to human brain responses, there was this correspondence between the NN layers and the hierarchy of the human brain. So a couple of papers which came out in 2016 and 17 basically showed that this gradient also holds not only in space, but also in time. So this was done with MEG. What this means is that the earlier layer corresponds to features that are encoded shortly after the stimulus onset or offset, and the deeper layer features are encoded in the brain, after, say, hundreds of milliseconds. And there was this, again, a gradient in time. And also the results for music recognition, I forgot to mention music, I said audio processing, so that the initial results were about music recognition, and it was shown that this can be done for speech as well. But now this brings me to the but part. So there's an if, right? So the results are quite easily reproducible if the data are open. And that of course, there are always exceptions, but that's most of the case. And the primary reason for this is that acquiring new data is difficult. But of course, I mean this for our particular problem domain. But I'm sure also acquiring data is difficult for other problems as well. But what I mean by acquiring data difficult in this setting is that, as I again tried to allude to earlier, we need very specialized data sets, which means experienced and motivated subjects who have to go into the fMRI scanner for multiple sessions, each taking hours. And I'll show you some numbers in a minute with optimized protocol. So it is really difficult to get it right, find the right subjects, so as to get a good quality data that we can estimate our models on. What this means is that most studies which kind of work in this domain nowadays, you analyze a handful of quite at this point in time old and pre-existing fMRI data sets. And the others who do not do this they rarely share their data. But of course, this is getting better, so people start to share their data more and more. And it's also kind of understandable because collecting such data requires a tremendous effort, and they want to kind of do as much as possible with the data to before I guess they share it with the community. And there was this small incident a couple of months ago. There was this Nature paper, I'm sure some of you is familiar, from the Gallant lab. So Nature, the conditions were that you have to share your data with the community, but then am I running out of time? Okay, but then they were like, well, after 3 years, they were still resisting, and they still haven't shared their data because of they had their reasons. But people are reluctant, and there are some data sets. Basically, I said that I'll give you some numbers, and these old existing data sets that most people use are around per subject. They collect 10 hours. So this is about 5,000 data set. Another data set per subject, 10 hours per subject, 5 hours, etc. This is good for reproducibility for everyone using the same data. Just one more minute. But not necessarily for original research because there's only so much we can do. And I would like to conclude with, so take-home message, we need more data for the next generation of studies to, we kind of exhausted everything that we could do with these already existing data sets. And for this reason, we recently collected this new open data set called the Doctor Who data, in which we put one subject, our master student, into fMRI scanner for 30 plus around 40 sessions, around 40 hours. It took 6 months to watch around 40 episodes of Doctor Who in the fMRI scanner, and this is a full brain multiband data protocol, so around 120,000 volumes. And the paper is currently under review, and once it gets accepted, the data will be shared with the community. And with that, we would like to just basically address all the remaining questions that we had that we couldn't with existing data sets. Yeah, thank you.
guess Christian should maybe think about setting up, and we'll have a question in the interim. Daniel.
You didn't make them watch any of the new episodes, did you?
No. We started with. Sorry, where's the question from?
Okay. Now, apart from the commentaries on Doctor Who,
we started with the 10th Doctor, so David Tennant, and 40 episodes from that. So not the most recent episode.
So you can graphically imagine what he saw. Tom, carry on, then you can have a question as well.
So it's really interesting to highlight this problem that what you folks are really best for is more data. And I was just wondering to what degree would multi-site or multi-center collaboration have a way that you can achieve this? Because if you're all sharing data with each other, then it's intrinsically open to begin with.
So I think that's a great idea, but no, unfortunately. So
what do you think are the barriers to that?
So there are two. So I guess the problem is the people who are collecting the data and analyzing the data tend to be separate groups. So we do both. We try to do both ourselves. But since the data collection people and then the people who try to fit these models to data are different group, I think they talk not as much as they should be talking. So kind of, hindering this, not just inter-institutionary, but also between group communication and hence such a solution, I suppose. But that would be excellent.
Okay, in that note, I think we can finish and. Move on to another great question of our time to be addressed by Christian.
Thank you. Thanks for asking me to participate. And again, thanks for selecting the title from me. From statistical to biological validity. So I'm not going to talk about FSL, because a lot of it has been said already, and anyways, it's not that interesting, despite the fact that we just released version 6, which is really cool. Much of what I'm presenting today is going to be examples of how in the group we're trying to actually work towards biological validity in the estimates that we're generating, and much of it is inspired by the discussion within the neuroimaging field through starting with the Ed Wool paper on double dipping and now the Ekland paper. It seems that methodologists have a brilliant time cutting each other up, coming up with silly mathematical statistical arguments about one is better than the other. And the real problem about the replication crisis in neuroscience, in my books, very often does not lie within the method section. It actually lies within the discussion section. It lies within the fact that many neuroscience papers are completely kosher within the method sections, estimate stuff correctly or use software correctly, but then completely extrapolate broadly, widely, massively within the discussion section, so that effectively the paper has very little to do with what you actually read in the method section. And it's made worse by the high impact publication systems now, where in Nature Neuroscience and Science and PNAS, methods and the exact description of what has been done has been relegated to the 0.6 font section or the supplementary material, which simply does not help reproducibility or replicability. So what I wanted to talk about from statistical to biological validity is a set of examples of typical neuroscience problems that I think could actually be tackled very differently if you were to start thinking about whether something is biologically valid. Prime example is that of parcellation. Parcellation, finding the basic organizational principles in cortex, let's say, is a very, very common problem, and many, many papers have tackled this. The vast majority of papers start with what I would call a bottom-up parcellation. So they start parcellating the whole brain, starting from a voxel. Now, if you look at it, a voxel is not a biological property. It's not a biological entity. It doesn't have any validity. It's forced upon us by evil MR physicists that cut up our data space and discretize. And in fact, I mean, In the red corner, wait a minute.
We've run out of time.
But you see, right, it's not even a quiet in voxel space, it's a quiet in frequency space. So the voxel is an arbitrary concept, yet the vast majority of tools and techniques for automated parcellation rely on the voxel as the underlying building block. And to us, that means that there's a distinct lack of biological validity, and that leads to many problems, like how do you scale, how do you choose the scale? At what point is it appropriate to stop parcellating? Right? And why do you think that when you start with an entity that you realize has no biological validity, when you start lumping them together, magically at some point biological validity will emerge? It's not going to happen. So you could actually start rethinking this. You could say, okay, what if we actually did it the other way around? So this was actually the PhD project of Eric van Oort, by and large, where the idea basically is, okay, we know that the brain at least is a biologically valid entity. How do we optimally split, kind of define subunits that retain some kind of level of validity? I'm not going to go through the algorithm, but what I'm going to show you is that this now leads to a parcellation that becomes somewhat biologically meaningful. So for instance, when we take the M1S1 complex and apply this algorithm, it will give us multiple possible parcellations at different scales, and we can actually see with this up and down what parcellations have highest cross-subject reproducibility, and you'll see that they come in triplets. So the algorithm is telling us that, okay, you can force me to kind of give you another split, but if I do so, if you force me to split, let's say, left sensory motor system into another parcel, I also want to do it on the right and in the center, right? Which is biologically meaningful because we know that that is a reflection of how the motor system is organized. Similarly, we can start looking across modalities and start looking into organizations. So this, for instance, is an ICP parcellation of the human thalamus on the left. In the middle, you see what you get from a post-mortem staining of 3 thalami using simple tracing, that's the staining studies. So this is post-mortem only done in the right thalamus. You can look at dice overlap, or you can compare what you're estimating from a parcellation like this, in vivo in this case even task fMRI, not just resting fMRI, against the only known atlas of the entorhinal cortex. So looking for kind of correspondence across what's already been published is an important feature that we shouldn't ignore, and we shouldn't just focus on, oh, do I hit my p-value? Here's another example of how you can actually use alternative types of data in this case, how you can achieve convergence across modalities. So what we've done there is compare the parcellation that we're getting with the ICP type of parcellation at different levels of granularity in trying to explain a completely different data modality. So here we've used things like dopamine transporter SPECT data from Barcelona. And we just check against existing atlases, the Harvard-Oxford Atlas and HO, or the Oxford-Imanova Atlas, OI, how much variance one type of description actually explains in this complete separate, as of yet unused type of data modality. And it turns out that we do effectively at low levels of granularity, up to 4 or 7 parcels, we do up to twice as well as you do when you use standard anatomically defined atlases. Sometimes when you look across modalities, you can also kind of get towards the point of divergence. And to illustrate that in an example, I've starting here from the Fallman and Van Essen diagram, the probably most famous description of what the early visual system looks like. And Malcolm Young, a year later, has actually used that data and applied multidimensional scaling to argue that it organizes along a dorsal and a ventral stream. Now, what Kuhn-Haake and the group has done is actually say, okay, all of this effectively starts from something like the COCOMAC database, which is antro and retrograde tracing data in the macaque monkey. So it's anatomical connectivity in a different species, in order to make statements about the functional organization in humans. So what if we actually go to functional organization in humans? If the starting point now becomes the partial correlation matrix from functional observations in humans, in this case HCP data, and the rest of the pipeline stays the same, so we use multidimensional scaling like Malcolm Young has done, in the HCP data, indeed we see divergence across modalities. What we see is that under this type of analysis, we identify 3 streams, a dorsal, a ventral, and a lateral stream. And we do that with high cross-day reproducibility. We have a very clear kind of differentiation between the streams. The within stream versus the cross-stream functional connectivity differs massively. It differs by 6 sigmas. So for those of you who were excited by the Higgs boson being defined by CERN or identified at the CERN, that has a statistical level of belief which is 5.5 sigmas. So we are about an order of magnitude more confident that the visual system has 3 streams than the physicists at CERN are about the existence of the Higgs boson. But how do you square this then with kind of what is like, I mean, it's like a truism, right? A Laminar diagram. Well, we would argue that we can explain that divergence because, or we would trust this divergence because we are closer to the objective of what this kind of study is about. We use functional data to make a statement about function in humans rather than structural data from a different species. And indeed, if you now look it through the literature, you'll actually find other papers in 2013, for instance, that already describe evidence for the existence of a 3rd stream. Now, to talk a little bit more about the classical approaches that we statistically use for arguing for validity. Here's an example where we use a new technique called connectopic mapping, and it's motivated by the fact that when we look at the functional organization, for instance, within, again, the sensory motor strip, we know that cortex has functional variation. I mean, Martijn van der Heuvel has shown that in the 2010 paper, for instance, when you do a seat and you place that from the medial wall and you traverse the M1-S1 complex from medial to lateral wall, you see a corresponding peak functional connectivity traversing in the opposing hemisphere. So there's basically organization within organization, right? The sensory motor complex has a hand area, and within the hand area we can even boil down things down to the individual digits. What we also know, however, which is not captured by any type of current analyses, particularly not captured when you look for parcellations, is that cortex has functional multiplicity. All the sensory modalities, whether it's vision, whether it's sensation, whether it's audition, we know that we can actually elicit multiple modes of organization. In the visual system, we can do high eccentricity versus foveal presentation, and we get a map that looks like this. But at the same time, we can show you different black bars, and then you get an orientation that looks like, or an organization that reflects orientation selectivity. Now, when you actually start modeling this sort of thing, using a parcellated approach, as typically is done, when there truly is a gradient, but you start enforcing a hard parcellation, you will get spurious clusters. And you will get them with high reproducibility and high replicability. Who knows the difference between reproducibility and replicability here? Okay. For those of you who don't, reproducibility means that if I give you my software and my data and the entire procedure, you can run it again on that data and you get the same results. Right? Replication means if I tell you the idea of what I was after, you acquire your own data, maybe you write your own scripts, you write your own software, you get an answer that corresponds to what I'm doing. Right? So these are very different questions.
But here you see that this is a very simple example where reproducibility and replicability just fail you. And indeed, if you have overlapping gradients that exist, you will observe in the data something that looks like this. So again, if you enforce a hard parcellation on this, in the presence of overlapping gradients, you will get wrong clusters with high reproducibility and high replicability. So what I'm arguing here is that statistical quantities that typically are being used in all these papers to argue that I'm better than you, are not necessarily all that useful if what we're after is biological validity. So in order to overcome that, we've developed another technique. I'm not going to go into that detail, but then what we do in order to get biological validity is to look at convergence again. Convergence across species, for instance. So if we apply this to the human striatum, on the left you see a speculated gradient that exists on the basis of a meta-analysis of about 68 monkey papers. Haber and Knudsen have published that in 2010. We can now see this gradient in vivo, in human, using resting fMRI, including its projections to orbitofrontal and medial frontal cortices. We can start quantifying it in terms of how robust can we do that within an individual subject, right? But crucially, we look for other aspects of biology or behavior. So for instance, we do brain behavior mapping. We show that this gradient loads onto behaviors that we naturally associate with the striatum. Or we go the molecular end. We show that it's biologically relevant because we can map it onto the genetics. We can show that these gradients are heritable. We can also show that these gradients are not just heritable, but we can show that the heritability in a twin study, the HGP is a twin design, the heritability is actually pleiotropic with the behavior. That is, the same twin pairs that drive heritability of the behavior drive heritability of the gradient. Right? Another aspect of biological plausibility. And ultimately, we then try to look at very specific predictions that we make. So for instance, when we look at this striatal gradient, we again look for convergence across modalities. So we show that a gradient we can estimate from resting fMRI maps onto dopamine transporter availability, for instance. And that then gives rise to predictions. In healthy controls, you would predict that it maps onto compulsive behaviors. So we go back to the HGP and identify the compulsive behavior. We look at only the 30 subjects that drink the most, or we look at the 38 subjects that are the heaviest smokers, or we look at the 63 subjects that have clinically significant symptoms of depression. And in that healthy control sample, find these associations that we will predict if this was truly reflecting underlying biology. And then in a patient sample, we look at how well this predicts, for instance, in a Parkinson cohort, how well it predicts UPDRS baseline symptom score. So with that, I wanted to kind of illustrate how we need to kind of look for convergence across modalities within systems that become biologically meaningful and transcend these mere numbers that we get from looking at our p-values and all these quantities that are so popular amongst the methodologies that are looking for reproducibility or replicability. And these are all the people in the group that have helped doing all this work. Thanks very much. I think while Robert's setting up, we can have a question or two.
Oh. Thanks, Richard. I like that very much. Coming back to the dorsal and ventral streams, I don't remember Leslie Angelider and Mishkin ruling out the possibility of a third stream back in the days in the 1980s or so, where they came up with the two-stream identity.
That's correct. They never explicitly said that it's only going to, but if you now look at neuroscience teaching, even at the master's level, the where and what pathway is being taught as a neuroscience truism.
Yeah. And I think you're quite right. I think we should. 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 I think
be constantly skeptical of this.
Yeah, it's a classical example of how any kind of hypothesis, scientific hypothesis, needs to be scrutinized continuously. If new data becomes available, we need to kind of try and see what happens if you run it on new data. HEP wasn't available when Beckmann and Finesse did their stuff.
Peter, and then.
Yeah, you discard a voxel as biologically relevant, which I can imagine. But of course, the issue of biological validity is at the level of the brain itself also not trivial. That's, for instance, if you take the Umut and Güçlü's postulation on radiocyte architectonics, it's a different postulation than when you look at the receptor architectonics. And you can look at the myelin architectonics and so forth and so on. Now, the question is a bit, which of these levels of anatomical organization are the real relevant ones for the computational characteristics that drive my functional postulation? And I think that's still
that's completely open. Yeah. Indeed, what I didn't talk about, if you actually look in very detail at the parcellation that we get into the sensory motor system from this ICP algorithm, you'll see that in the superior areas it maps very, very well on the Zillis and Amund's atlas. So it maps onto psychoarchitectonics. And we know from functional observation that there should be kind of a horizontal granularity along the lateral part of the sensory motor strip. But if you look now into the psychoarchitectonic atlas, that is not what they're reporting. Psychoarchitectonically, right, particularly at the level of the hand area, it becomes a long superior-inferior strip. So while in the top portion we find a very good match, in the lateral portion of the sensory motor system, our atlas deviates significantly and maps much more closely onto what we know from functional activation studies.
Okay, I think on that note we will move on to Robert, who will tell us open science and or team science.
Yes. So this is something like in a very short discussion with Tim when he stepped into my office, in discussing what I could contribute. This is the title that we came up with. But what I want to do is very shortly give an opinion, give my views on something that we've recently been discussing a lot within the Donner Center, which is open science, but it's also team science and how these two might relate to each other. And I realized that when reviewing some of the recent publications that came out from colleagues in my center, there's some characteristics that I found standing out. And so these are papers that I picked randomly from PubMed from colleagues. And rather than specifically zooming in on papers of colleagues, I just want to zoom in on the principle. So what I did is I just Googled for random names. There's actually a website where you can generate random names. So I came up with a random publication. It's not totally a random topic. But what you can see is that, well, if we would, at the Donner Center, if we would have such a publication, of course we would all be very happy. Publications are always good, but there are some other characteristics here as well. And one of those characteristics is that some people will be more happy. And that's typically, if you think about the gradient, you have the horizontal gradient. So the first and the last author are typically the most happy. And the ones in the middle, well, they're also happy, but not to the same extent. And a second feature that I realized when looking at these recent lists of publications is that there's this small star, which I will also come to. I think that doing high-impact science is difficult. It requires more Teslas. It requires more expertise. But I realized that the number of contributors on publications is actually only growing slowly. So the publication that I showed you was 5 or 6 authors. Some of them have 3, which I think is pretty average. So this is a study, a meta-analysis of 24 million papers that appeared on PubMed. And what it shows is that in 2010, the average number of authors on the publication was about 5. And if you just look at how this is increasing, then it doubles every 30 years. So 30 years ago, it was 2.5. Now it's 5. And I think we're pretty much in an average range. The second thing that I realized, that although the papers mention a corresponding author, in my experience, so this is my personal experience, there happens to be a very little correspondence following the publication of a paper. Like the correspondence is usually, well, we eat pie at the coffee machine, everyone celebrates, like we have a short celebration. But it's not that we have a lengthy discussion on the content of the paper, it's rather we're happy that the paper appeared. And that is something that I see as being like not necessarily aligned with these recent discussions that we've had within the center on doing more team science and doing more open science. And I totally have to admit that this is not the only examples that I found when I Googled for recent publications of the Donors Institute in PubMed. This is one example where I even had to scroll down to the last page of the paper, where the list of authors was actually like mentioned more or less as an appendix, in the same way as like sometimes a method section is being pushed away. And this is another one where also a long list of authors is mentioned in very ambitious and large-scale studies. So these also exist, but I think they're not so common. Of course, a lot of you know that I'm very much into open science. Like I've been sharing research methods for a long time in the form of the Field Trip project. And recently I also started working on, together with colleagues here at the Donors, on the Donors repository, which is on sharing data. We have a donor section on GitHub, and that's also something that we're recently starting, like promoting more with the Friday afternoon hackathons, like training more people the skills of like what is it if you do a pull request. I think that in 1 or 2 years from now, everyone should just feel comfortable with that as a way of contributing to open science. And of course, on a very like non-technical scale, we also have a lot of efforts which are on open communication, like the Donors Wonders blog, which I think is really an excellent example of how we can like be more open towards the audience about our scientific findings. And now Tim came out with something totally different, because Tim is actually also really enthusiastic about the things that he developed. And what I found interesting in reading his thesis, that already in chapter 1, he is referring to methods and data being publicly available. And it's only in chapter 5 that he actually starts mentioning, so where are these availability of the data and the methods? So I think that, so this is my personal opinion, I think that if you want to move more towards open science and towards team science, we should actually consider not only considering the happiness of the first and the last author, but actually also weigh the contribution of the middle authors more. We might want to make more middle authors part of the study, so that the study is actually a team effort, and that the contributions of the team as a whole are being rewarded and are being seen as relevant. And furthermore, I think that if we want to see open science and team science contribute more to scientific impact, we should also consider the audience. And I think the audience, or the users of a method, the users of a data set, the people that want to replicate, want to reproduce and replicate a study, I think those are very important in our efforts into making more scientific impact, because that's what I think that we should try to aim for. So I think we have more people to involve in the team, not only the people here within the donors, but also the people outside the donors that are basically our stakeholders. So I just want to put up some ideas. So I think that achieving excellent interdisciplinary research, which is the mission of the Donors Institute, requires collaboration in skilled teams, it requires the right tools, and it should consider all the stakeholders. I think the audience that we're doing this for is not only there to upvote the measurable impact, which is of course where the first and last author tend to be happy with, but the audience actually should contribute to increasing the long-term impact of our scientific findings. And I see team science as a very important way to increase the cross-disciplinary expertise that we need to do for these like very ambitious studies. And I think that open science is needed to involve more of the stakeholders in the studies that we're doing. So that's the audience, or that's the users. So what I'm arguing for is that we should involve more of the audience more often in our science and actively work together with them to increase the impact of our science. Okay, thanks.
after that, we should have an active discussion. I think Tom has got a short head first.
Great. So, I think that was an excellent and very inspiring talk. So, with the exception of the distinguished gentleman over here in the corner, you're speaking to an audience that looks like about 100% early career researchers, right? So, I see like this is very important to target this because some of us are going to go on to kind of be with these folks in follow at some point. But like, to what degree do you think that the very important things that you're talking about are not happening right now because there's not a will amongst PCR's early career researchers to do them? And to what degree do you think it's actually not happening because of the incentive structures created by journal editors and funding bodies? And if, as I suspect, the answer is going to be option B, what do you think we, as the early career researchers in the room, can do about that second problem?
I totally agree with you that I see option B as the thing that is the most challenging, like the incentive structure. Of course, a very important incentive structure for early career researchers is to obtain a PhD title, which of course we hope that Tim is going to do today. But in that process, it is quite a challenge, and that the challenge with the promoters, with the supervisors, to also allocate enough time to do things which are not measured along the normal way of measuring scientific impact, like the old-fashioned way of measuring scientific impact. And as such, I was very, like, one of the things that motivated me was seeing Tim's publication list in the back, where he had quite some publications where he was like in the middle position. And of course, Tim also having published his methods prior to actually publishing his key scientific findings. So I think it's this incentive structure requires an open discussion between the early stage researchers and the supervisors that influence where the supervisors influence also the criteria along which the early stage researchers are being measured, are being evaluated.
Right. Because I can imagine in a parallel universe, sort of talented researcher like Tim being told maybe you should focus more on getting high impact first author publications because that's what's going to fund your career going forward. That's what's going to make you attractive to funding bodies like NWO and whoever else. Obviously, that hasn't happened, and we as a community will benefit from those tools. Mm-hmm. But I definitely see some kind of tension there.
Yeah. Yeah, I agree with much of what you said. But how do you square this with things like the Vancouver Convention on Authorship? With all these, I mean, you've probably seen the recent discussion kicked off by Nighres of the most prolific scientific output, where he basically starts to shame everyone or a long list of 200 and something researchers worldwide that happen to have more than 72 papers accepted in 1 year over the course of
Yep.
There's a long list.
Yep.
And if you look through that list in particular, you'll find that from the Netherlands, for instance, it's full of people from Erasmus.
Yep.
Who end up on this list because of Generation R and all these large data efforts. Likewise, people that are active on the HCP will end up being invited. So I completely agree that we should be liberal with offering authorship, because for as long as our incentive structure is such that there is some kind of reward from being an author, we should be liberal and acknowledge all kinds of contributions. At the same time, there is the request also from journals to specifically identify what contribution an author has made, to specifically identify what portion they're responsible for, to request that all authors sign up on a publication.
But I think a very important aspect is that we're still considering our scientific output to be authored. Whereas I think that the scientific output is now the consequence of many people working together, many people giving a contribution. So I think that if we don't consider the people that are mentioned as a publication as contributors necessary as authors, we can also start thinking about like how do we want to credit the different people that have contributed to a study. That's of course some journals already make that more clear by categorizing authors in different. Or to reduce them to one author, basically say this is the donors team that has done this. It's about the incentive structure where this is problematic, because at this moment the incentives are primarily measured according to authorship. Like it's the people that are mentioned, like as the last on the paper, or the people that are the PIs on grants. Those are being rewarded for the grants that are being pulled in, or the. Yeah. Yeah. Yes. Yes, so I think the examples exist where we can learn from, but those examples are not necessarily yet embraced. Of course, we have momentum now, but of course, scientific culture is also sluggish. It's quite challenging to change the scientific culture, and I think we are in an excellent position actually to start making these changes.
Well, okay, you wanted to say something. I didn't.
Maybe a more comment rather than a question on this topic of dealing with authorship, especially when you have maybe complicated study where multiple people were involved into getting excellent data, and people who developed some tools, and then some other people who went afterwards did a completely different analysis, and everybody is in the paper, right? But a way to make it more transparent who's been doing what part of the project is indeed to use open science. Because if the people who collect the data then go ahead and bring the data to make it available as a paper, same thing for the software, etc. The people who realize afterwards only have to cite those publications. They help those people also by showing their work is highly cited, even if it's just putting data out there. And we'll also keep their publication focused on this is the only component.
Yeah, but I think this necessitates a better system for distributing rewards among the people that contribute to the important impact. Citing papers is easy. Citing software is already more challenging. Citing data sets is not necessarily trivial either. Of course, it is getting better, but that is something that we also actively have to work on making sure that the rewards for the work actually go to the people that deserve these rewards.
Okay. If there aren't any more questions or comments. Okay, now we get Horest later today, so thanks again everybody for a really stimulating session this morning, and hope to see many of you this afternoon. I think we're all on the list.
Are you going to go straight to the aula from there? Then I'm going to draw the speaker side.
All right. We've already got time, but we have to get the aula at 2 o'clock, right? We need to do the aula so this is on, right? I'll take the speaker. You'll get your family, right? I'll take the speaker.
I'll meet you there because I'm going to have to drive.
I was actually kind of your exactly somehow open site.
So this for me is really like this is I think the most important.
That's what I was asking. What are we going?
It seems to me that like in 2018 we're in an era where with like unparalleled specialisation in terms of skills, but we're still basically expected like to be homo universalis and have all the white. And it's amazing how much you see this in an institution like the institution now, and it's really like across all institutions, the attitude seems to be there's this new thing happening in science. It's very important that all the people learn how to do it in addition to all the other things they're learning how to do. So and I see that with the young people now as well. It's like we all have to we've all got to attend Symposium about how to share our data. We've all got to attend classes on how to safely share our data. And those things are super important, but it seems to me that they're actually really important at the institutional level, that we have people that can do all those things. And I guess that's where the team science is, right? That you use all of those people.
Rather than that people getting a PhD in medicine or PhD in biology, they should be getting a PhD like in academic writing, or they should get a PhD in software development,
data visualization, all of these kind of
all the individual skills, like the skills which are really getting more and more important.
No, I couldn't agree more. No, I think the thing that we have now as a consequence of this is that every early career researcher basically spends the majority of their time doing the things that they're least good at. Because it's like I know after 6 years of doing it, know quite a lot about how to program and build it, but don't know how to make amazing figures or whatever. So it's like I feel like I spend hours and hours of my time doing that, and it really seems like shouldn't we have somebody to do that? It's like if you work for a company, you have a department that is specialised in that. You have or you invite people.
Or you could say it's not about should we have someone to do that, but should we have someone to organise it such that this can be done.
To facilitate doing that. Right, absolutely. No, I couldn't agree more.
I'll quickly grab my.
Oh yeah, cool. I'll see you and then I'll join you.